Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the course on computer aided drug design. We will continue on the topic of target based drug design or related to docking. Let us again look at this protein data bank that is called PDB, which contains the 3-dimensional structure of large number of proteins. We also have ligands also present in some cases, and in some cases ligands are also not present. So just we will have a look at that. Okay, this is the PDB, it is the protein data bank. So we can do a search the PDB. If we know the PDB ID, we can do a search, for example 3V98. okay, that is 3V98, that is a 5-lipoxygenase. That is an enzyme in the arachidonic acid pathway which converts arachidonic acid into HETE and so on. So we can look view that under JSMOL. Okay, so this is your protein. As you can see, it's got a and B okay, you can see this protein, it's got both the A and B. Now it's also got an ion, okay, because lipoxygenase has an ion. So the ion is involved in the redox cycle, that way it activates the protein, and when arachidonic acid binds to it and gets converted, it gets deactivated and so on actually. Okay, so this is the protein which does not have any ligand attached to it. There is a again a 3V99, okay, that is again a 5-lipoxygenase. Okay, it has got a arachidonic acid attached to this. So we can, okay, so we, as you can see here, so this protein has arachidonic acid as well as the ion. So we can specifically look at where arachidonic acid present. Okay, so this is the arachidonic acid. So we can say view the protein from the arachidonic acid pocket. Okay. Yeah, so you can see that this is your arachidonic acid, it is a long chain hydrocarbon. You can see that protein, okay, and if you want we can even look at the ion that is present. As you can see here, this is the ion that is present in the protein, and with the corresponding amino acids that are surrounding there. So this picture shows how the arachidonic acid is interacting with the protein. Okay, what are the amino acids that are involved which interacts. So this is the arachidonic acid. This is the entire protein that can be viewed. We can look at another protein of interest, that is called, yeah, that you can see this, yeah. So you can see the arachidonic acid here and here, and the protein. Okay, so you can see the amino acids with which it is interacting. Now we can look at another protein that is called 5KIR. this cyclooxygenase 2 COX2 5KIR, yeah 5KIR. Okay, this is a COX2 cyclooxygenase 2, and we have the Vioxx. This is a selective cyclooxygenase 2 drug which is bound to that protein. Okay, so when they crystallize this protein, they put in this molecule and then they perform a crystallization. So the advantage is when we crystallize, we will know where this particular molecule is present, so we will know the active site pocket and so on. Okay, so this is a protein called a cyclooxygenase 2. So you can see there are a lot of ligands which are there present in this protein. So Vioxx is also called ruficoxib. So we can look at so we can look at how they are present. Okay, so we can look at how Vioxx is present, as you can see here. Vioxx is present inside this particular cyclooxygenase 2 protein. Okay, you can see it there here. So PDB is quite an useful protein data bank. It contains large number of proteins. It also gives you the sequence of the amino acids. Okay, sequence display for the entity in PDB 5KR. Okay, so as you can see here, it gives you the sequence of this. We can also do a sequence similarity, structure similarity. So we can do a sequence similarity also using this, as you can see. Sequence similarity, we can also do structure similarities. Entries in PDB 5KR, so we can look at the structural similarity. So this database gives you structures. So when we try to perform docking later on, we may have to download one of the protein of interest, and then we can perform the docking. Okay, we will look at docking at a later point of time. So let us go back to our presentation. Okay, so a protein and ligand interacts generally through non-bonded interaction. Okay, so we have looked at what those non-bonded interactions could be long time back. Electrostatic interactions, okay, because you have charge, so there could be some interactions electrostatically. Hydrogen bonds, okay, so there could be a hydrogen bond donors, acceptors. In the protein there could be hydrogen bond donor, that is the amino acids in the active site of the protein. There could be hydrogen bond acceptors, donors in the ligand. So there could be a hydrogen bond interaction. There could be a van der Waal interaction, okay, between the protein and the ligand. There could be dipole interaction because you have water, so many functional groups which form, and of course ionic interaction. The protein, there could be a anion cationic in the active site. Similarly, the ligand also could have a cation anion, so there could be ionic interaction. So all these are called non-bonded interactions. Unlike the normal covalent bond, generally the bond energies of these non-bonded interactions are very low when compared to that of the covalent bond. But these bonds keep the ligand in place and keep it in the proper conformation for inhibition or conversion to happen. Okay, if it is a lipase catalase reaction or enzyme catalase reaction, you are going to have a conversion. Okay, so basically what happens, we have the ligand. This is just a pictorial. There is something called an active site, or there could be some other secondary site, allosteric site, minor grooves, major grooves. So the ligand goes and binds to that and forms a ligand protein ligand target complex. So, if you look at thermodynamics, ln k is equal to minus delta G 0 R by RT, delta G 0 can be written as delta H 0, okay, and T delta H minus T delta S. So, the T here goes away, you have these two terms. Now, this K equilibrium constant is complex divided by ligand into target, okay, concentration, okay. This you must have studied long time back in thermodynamics. So, this is very standard, okay. So, from delta G we should be able to calculate K from this equation. Okay, so if you look at covalent bond, the strengths of covalent bond, as I said, they are quite large. CO single bond, CO double bond, look at these energies, and again look at the distances. So, if C double bond O, the distance is going to be much smaller than C single bond O. Similarly, C single bond C, you have 86 kilocalories, length is 1.54 angstroms, C double bond C 143 kilo, big jump, 1.33 angstroms. They come closer and closer, okay. So, covalent bonds are characterized by high bond energies, short distance, high direction. The direction is very focused and fixed, okay. So, it is not very random and as you feel like, whereas hydrogen bond for example is not so directional. So, that is the difference between a non-bonded interaction and a bonded interaction. So, if you look at the non-covalent interactions, we looked at covalent interaction, this is non-covalent hydrophobic interactions, okay. That is hydrophobic groups are there, right, CH3, C2H5, the energies are very little, less than 10 kilocalories. Look at these numbers, 1 to 20 is electrostatic, hydrogen bond 2 to 30, pi aromatic stacking 0 to 10, van der Waals 0.1 to 1. So, whereas look at these numbers, big numbers 80 to 160, whereas these numbers are 1 to 20. So, non-covalent interactions are characterized by low energies, poor directionality, direction is not very fixed unlike the covalent. Of course, the hydrogen bond has some directionality because you need to have generally a O or a N forming a bond with the H of OH or H of NH. So, it has got a directionality attached to, whereas other bonds like electrostatic or aromatic stacking, it does not have much directionality. Salt bridges, when oppositely charged groups in proteins are within 5 angstroms, okay, anion, cation, cation, anion, they are generally referred to as ion pairs or salt bridges. So, look at the energies, they are quite low, ion pairs on the surface less than 1 kilocalorie per mole on to this for stability, buried salt bridges greater than 4 kilocals per mole to be stable. So, numbers are quite low, okay, very low. So, ion pairs form salt bridges. Let us once again look at these non-bonded interactions, although we looked at them in molecular modeling, once again we will look at. So, the energy is given by some constant, okay, Z1, Z2 charges, E the amount of electrical charge on an electron, that is 1.6 × 10 to the power minus 19. Epsilon is the dielectric constant of the medium that comes in the denominator, or is the distance, okay. The ion pairing interaction, it can be an attractive or a repulsive force. There are long range interaction. I mentioned these terms long range when compared to short range, because you have R in the denominator, whereas if you look at van der Waals hydrogen bond, you will have R raised to the power 6, R raised to the power 12, R raised to the power 10 in the denominator. So, they are called a short range. Of course, this is dependent on the dielectric constant of the medium. So, dielectric constant can vary dramatically. For example, if you look at water 78.5, if you look at CdCl3 it is 4.5. So, big difference, okay, almost 20 times difference. So, this is taken from this particular reference. So, the dielectric constant of the medium can change the energy of attraction or repulsion between two charged molecule. Ion dipole interaction. So, you know what is a dipole. For example, water, HOH, so the H's are all forming plus, like a magnet, small magnet. O is forming minus, so it is like a small magnet with minus and plus. So, that is the dipole, and then you have the ion of certain charge. So, these are weaker than ion-ion interactions, long range interaction than ion pairing. Some directionality is there because the interaction is can be attractive or repulsive. Solvation heats closely correlates with ionic radius. So, this is the equation for that. So, we have say suppose we have Q1 and Q1, okay, which is the dipole plus, and this is the minus, and then we have a ionic, okay. So, the Q1 into Q2 L is this distance, cos theta divided by 4 pi epsilon 0 r square is called the ion dipole interaction. Dipole interaction. So, you can have two dipoles, they are very weak, they are longer range interaction. Now, you have r raised to the power 3, some directionality is there again, okay. It is called a dipole interaction. Of course, van der Waal interaction you all know, right. Polarization when a charged molecule or a polar molecule gets close to another, mutual changes of the electronic wave function strengthen the interaction. So, we can have ion induced dipole, dipole induced dipole, dispersion, all these. So, they are very short range because you have 1 divided by r power 4 minus 1 divided by r power 6. Of course, A will be there, B will be there, exists between all atoms and molecules. So, like this, you know, B raised to the power r power 12 minus A raised to the power r power 6 and so on. So, ion induced dipole will be like this. So, you have r power 4 coming here, the dipole dispersion forces will be like this, right, r power 6. So, this is called the van der Waal interaction. Then we have the London equation, that is weak attraction r power 6, and this is the repulsion between atoms at very close distance r power 12. So, we have different types of effect based on r, okay. For a electrostatic we have divided by r, for ion dipole interaction we have divided by r square, for dipole interaction we have divided by r power 3, for van der Waal London dispersion and then repulsion we have r power 6 and r power 12 in the denominator. Then comes hydrogen bonding. This is a very important parameter which comes forms when a hydrogen atom is positioned between two electronegative atoms, mainly oxygen and nitrogen, okay. So, we can have like this, you know, a donor with a H, a donor with a H and then an acceptor. These short range distances we are talking in terms of 2.5 to 3.5 angstroms, theta is 180, phi is 120, ideal angles, 40 to 15 kilocals per mole, that is strong, 14 to 4 kilocals per mole moderate, 4 to 0 kilocals per mole very weak. Several factors affect hydrogen bond: solvation, dielectric constant of the medium, hydrogen bond donating accepting ability, electronegativity of the donor, okay, HF greater than HCl, HBr greater than HI, acceptor water greater than H3, and that is an ammonia, H2S, H3 poor, but F is very poor. Charge, hydrogen bond may be strengthened or weakened by concurrent electrostatic interaction, okay. So, if concurrent electrostatic interactions are there, the hydrogen bond interactions can become weaker or stronger. Then we have the pi interactions. This is that benzene rings. Now, you can have two benzene rings. Charge distribution in benzene produces large quadrupole movement in the form of two dipoles aligned end to end. So, we can have cation pi interactions. This is much stronger. Cation pi interactions can be as strong as ion dipole interactions, okay. You can have different types of benzene rings, face to face interaction. This is very unfavorable because the surface is generally negative. So, one benzene ring have a negative surface, another benzene ring have negative surface. So, the interaction is very poor. This pi interaction is also called pi stacking. T-shaped interaction is the most favorable. That means one benzene ring is flat, another benzene ring is perpendicular like a T, energy is 2 kilocals, and of course this is the most favored. Parallel displaced, that means one benzene is flat, another benzene is on, not exactly on top, displaced but parallel. In water for large aromatic molecules, as it enhances also hydrophobic interactions. Okay, this is the most liked actually, especially preferred in water, and the T type of interaction is also favored, but this generally we may think one on top of another, that is the least favored because we have the negative charge. There could be a negative charge interact repulsion between two benzene rings. So, this is also a quite an important non-bonded interaction we need to consider. Then we have donor acceptor interaction occurs between two molecules with respectively a low energy empty orbital acceptor and an high energy filled orbital, that is donor. Okay, so it gives you the electron into the acceptor. Insertion of a non-polar molecule in water causes an interruption of the loose hydrogen bond network. Then the water molecules are forced to form hydrogen bond only with outer molecules, creating a cage around the solute, right. If you put in a hydrophobic molecule, there is a cage like leaving the hydrophobic molecule and the other water molecules start interacting. Okay. Association between two hydrophobic molecules lead to a decrease of the number of water molecules trapped in the cage formation. Okay. So, if you have two hydrophobic molecules, they start associating with each other, so water molecules get moved out actually. Okay, they are not trapped. So, when we do target based drug design, and we need to know have a compounds library, we need to get the protein target we can get from the PDB, compound library. I showed you so many compound libraries, we can get the mol2 file or SDF file of the molecule, we can get the conformation, minimum energy conformation. We need to know something about the binding site of the protein. So, because a protein can have many sites, a substrate based site, active site, allosteric site, minor groove. So, we need to know which site the ligand is going to bind to. So, if you are doing a competitive inhibition, so obviously then the ligand has to be competing for the substrate of the protein. If you are looking at allosteric non-competitive, then you look at other sites. So, deciding on the site also tells you what type of drug you are planning to design. Then you dock means the ligand goes and binds to the active site, forms a non-bonded interaction. We have the non-bonded interaction energies. This depends upon the various conformations the ligand can take. So, if the ligand is very flexible, it can take different conformations into the active site. So, it can take the most favored conformation so that it goes and fits nicely into the active site based on the various non-bonded interactions I talked about. So, a very flexible molecule can take different conformations, so it can go and fit into nicely the site. Okay. Then many softwares perform a scoring, it calculates a number, okay, based just like your marks, it calculates based on the energy of the non-bonded interaction, that is the binding energy, okay, or interaction energy. So, many softwares have different ways of calculating the scoring function. So, higher the scoring function, then binding we can say is better. So, if you have large number of molecules, you keep binding and then look at the scoring function and then say these molecules have very good scoring function, these do not have good scoring function. Or if the software does not produce scoring function, it also gives you the binding energy. So, more negative is the binding energy, we can say better is the interaction. We can also look at other interactions, how many hydrogen bonds it has formed and so on, and then we can conclude that certain ligands have good binding to the active site, certain ligands have poor, and then select the preferred ligands. Okay. So, you prefer ligands which you think bind very well, then prepare a list of prioritized compounds, maybe you start synthesizing them and then start studying the experimental activity. Of course, in between you may also have to look at Lipinski's rule and other ADME rules to see whether those molecules satisfy the drug likeness property or biocompatibility property. We looked at so many different rules, so we can also shortlist molecules. So if you have say thousands of molecules, we can either do the Lipinski's rule and ADME rules first and shortlist molecules, then perform the docking and then select the top 10 or 20% of molecules which have very good docking score or bind very well with high binding energy, and then take them for experimental synthesis as well as experimental biological evaluation using cell line or whatever biochemical assays which you have planned. This is how the target based screening is carried out actually. So we can combine both the structure based, that is the ligand based screening, and target based screening and ADME rules screening all together when we talk about in silico drug design. Okay, so what do? We start with look at the PDB, protein data bank. Like I showed you, it contains more than 50,000 non-redundant protein structures, but then it is only 1% of the 5.5 million protein sequence in Swiss prot. Okay, so the Swiss prot is a web database which contains sequences of protein. That means it is a 2-dimensional sequence, whereas PDB contains 3-dimensional structures. So that means a lot of proteins have still not been crystallized and their 3D structures have not been obtained. So there is still good opportunity for us to do a crystallization study and get the details, 3-dimensional details of proteins. Okay. So let us look at this Swiss prot. Okay, so this is Swiss prot or UniProt, which gives the gives a sequence of a large number of proteins. Okay, and then we can submit them and compare them. Okay, so okay, so the 5-lipoxygenase, the sequence is given actually. Okay, sequence of 5-lipoxygenase is given. So it is trying to look at proteins, different 5-lox proteins, and trying to get their sequences, that is the 2-dimensional sequences. Okay, this I got the UniProt number that is called, okay, yeah, this is one, the UniProt number of 5-lipoxygenase, that is human 5-lipoxygenase, is called P09917. Okay, the job is still running. So we can look at various proteins, 5-lox proteins, which has a sequence similarity of this particular protein actually. Okay, it is still running. It will take a long time because there could be a lot of proteins of 5-lipoxygenase this is from human which we got, yeah, this is from human which we got. Okay, there could be many proteins from different species. Okay. Okay, so what it means is there are only 50,000 proteins, 3-dimensional structures in the protein data bank, but this Swiss Prot has almost 5.5 million protein sequences, that is a 2-dimensional protein sequences, which is only 1%. So, we still do not have the 3-dimensional structure of many proteins. Okay, and that this number is only a very small amount of proteins on the earth. That means there are about 5 million organisms, each organism has 5,000 genes. So, that is a very small number. That means we do not have information about large number of proteins, almost 99.99% of proteins found in the earth we do not have actually. Okay. So, we will not find the 3-dimensional structure of many proteins involved in many diseases for human. So, we may find for other species. So, we need to perform something called a homology modeling. That means we, from the protein sequence of say human related protein, we try to see whether any protein 3-dimensional structure of protein is available in PDB, and then we perform a homology model and get the structure. Okay. Okay. So, target based drug discovery is knowledge of structural biology, functional genomics, chemical biology and so on actually. So many areas combined together, we need to have some knowledge to get the target based drug design. Okay. So, if you again go back, let us see whether the job has been done. The job is still running because there are too many proteins. So, we will continue more on the target based design in the next class as well. Thank you very much for your time.